Hi there, my name is Bruce Nash, and I work at Cold Spring Harbor Laboratories DNA Learning Center, where I'm the Assistant Director for Science. Thank you for joining me today for my presentation, which is going to be on tracking the pandemic with viral strains. I'll go over some basic information, and then I'll go into some tools that you can use to look at the virus DNA, RNA actually, and get a sense of what scientists do when they're trying to work with the nucleotides, the sequence from a virus. So I'm going to start out my presentation now. I'll be going from the presentation and then to some online tools, which I hope you'll enjoy. Although it is going to be quite a bit, and I hope it works out well. Okay, so once again, welcome. Okay, for those of you who don't know what the DNA Learning Center looks like, here's just a picture of the DNA Learning Center, which is part of Cold Spring Harbor Laboratory. It was originally a small schoolhouse and has been renovated into our Genetics Science Center. Okay. I'm sure everyone's aware that we're in the middle of a global pandemic called COVID-19, and that this started in China in, it looks like, probably November or December. So I just want to remind you, although it seems like a really, really long time, the first reports of this virus came out in December, and it was on December 31st that China let the World Health Organization know about the virus. So here's just a shot from the South China Morning Post from New Year's Eve, referring to a suspected site where the virus could have come from, but it seems more likely now that it just spread from there through the initial patients. But there were already cases that we didn't know about that were outside this seafood market. As of today, by looking at Johns Hopkins University's pretty ubiquitous site, almost 2.5 million cases have now been confirmed, and they're spread throughout the world. The only place that hasn't been touched yet is Antarctica. Okay. So this is a very fast pandemic. Just to give you a comparison, it spread very quickly throughout the world, much more quickly than SARS did. And, in just a few months, it went from just in China to everywhere, as I think we all know, okay? Kind of remarkably, the science surrounding this pandemic has been moving very quickly. And that's in part because DNA sequencing technology has come a long, long way. And now you can do DNA sequencing very, very quickly. It took a little over a month for the genome, the RNA sequence of this virus, to be published. And that's very fast. Okay. Part of why it traveled so fast, apart from us being in a very modern society where people move around a lot, is that it's a very infective virus. And you might have heard about this a lot. Most estimates range between 2 and 3 people being infected each time one person is infected. And that's about the same as some other famous viruses, including HIV and SARS. But there are also some newer estimates by a large group that suggest it might be as infective as polio or smallpox, which were very infective, but not quite as infective as measles, which is very, very infective indeed. So, the cases grew very quickly, and in different places, they've undergone really spectacular growth. you know, going from the first case in a place to really big numbers seems to take 2 to 3 months. And depending on how those societies or places react, the rate of infection and the spread can vary quite a bit. And of course, in the United States, and especially in the New York City region where I'm at, there have been many, many cases. And this has been proven to be quite a challenging virus indeed. So in case you haven't seen it, here's a picture of some virus. This is from the first patient in the United States. So this is an electron micrograph of this virus, which is now called SARS-CoV-2. Okay. And what I want to tell you about today is just a story about this virus, but you know, focusing on the genome and some of the things the genome can tell us. I can't tell you about everything. I can't show you everything. And in fact, I'm not a co-virus specialist. I'm a geneticist, but this isn't really my area. So I have to admit, I couldn't tell you everything about this because I'm not really a complete expert, just so you know. Okay, so to go back a little bit, you know, I'm talking about a genome. Well, what is a genome? Well, a genome is made up of DNA, or in some viruses, out of RNA. Okay, so here's just a picture of Francis Crick, one of the proposers of the model of the double helix of DNA, and also a very important player in understanding how DNA and RNA and protein all relate to each other. Okay, and just to remind you, in organisms, DNA encodes information that goes through an intermediate RNA, which is then read by an organelle called the ribosome into proteins. And proteins perform many of the functions that are important for living organisms, and they're related little things, viruses. Okay. And the genome is all the hereditary information encoded in an organism or a virus, which are not considered organisms, but they have many of the same aspects as living organisms, including having a genome, which is either made of DNA or RNA. I've just put in a picture of a human genome. It's a picture from a site called NCBI, the National Center for Biotechnology Information, which I'll be showing you a lot of today. Okay, so the human genome is pretty big. It's over 3 billion nucleotides, or building blocks of DNA. And it has many, many different genes in it. And I just went into a genome viewer, which is part of NCBI. And I went to one region just to give you an idea that, you know, in the DNA are sections that have functions. These are genes. And the functions are often to code for proteins, although there are other genes that code for RNAs, which I won't talk about today. And right in this little region, there's a deep green square. That's a gene for a receptor for a bitter compound that we sometimes do an experiment on. That compound is called PTC, and this receptor is just one example of a gene, and it's in between some other genes that code for different proteins. Okay. Now, the human genome has many, many genes coding for many, many proteins. I think the latest number is in the range of 18 or 19,000 proteins. And then there are lots of other genes that code for RNAs as well. Okay. Well, that's all great. But, you know, we've got this viral genome from this new virus. It's a really threatening thing. And of course, scientists wanted to figure out more about it. You know, where did this virus come from? How does it work? What can we do about it? And one of the keys to understanding all of this is to look in this DNA, in this RNA, because it's an RNA virus, and figure out what the genes are in there and what they might do. There are also other things we can find out in there. But to do anything with this, we have to take a look at the sequence and do some sort of search with that sequence to see if we can find anything that makes sense. Okay, so scientists, much like anyone else, we use computer tools in a field called bioinformatics to look for sequences and figure out what they are by looking for other related sequences. Okay, so we need to do something a bit like an online search in a browser or a word search with the genome to see what we can see. Okay, so to do that, I want to go to a website that's part of NCBI. It's called NCBI Nucleotide. It's a database that you can search for things in. Okay, so I'm going to go to a different window now. I've put the link in here just so you can get a sense of that. But I'm going to go instead to another site, and then I'm going to go into this site called NCBI Nucleotide. You'll be able to find it very easily. You can either go to NCBI's homepage, or you can do a search for NCBI nucleotide in a browser to use one search to get to another search. And then in here, I just need to search for something that makes sense. And we know that this started in Wuhan. So I'm just going to do a search for Wuhan virus and see if NCBI has any suggestions for me. I have to make sure I spell that right so that it works. Okay, so here we go. I'm going to see if typing in Wuhan virus, just based on that one news article that I had in a slide earlier on, will work. And what happens is, NCBI nucleotide, it has a pretty good word search, and it gives me what I want, which is really nice. It's a pretty powerful tool. And if you use the right keywords, you usually get something close. You might have to dig around if you're doing a different search. Okay, so here I am. I found severe acute respiratory syndrome coronavirus 2 reference sequence, or genome. This is the published first genome, which sort of defines this thing, this virus. Okay? And immediately, I see some cool things. I can see that, it's making some things. Here's this view of the virus. It's making something called a polyprotein, something that looks like a shorter polyprotein, and then some things called SORF3A-NE. Well, right now you don't need to worry too much about all those details. Needless to say, these are different products that are encoded in the viral RNA. They make proteins that are important for the virus, and I'll come back to them a little later. But for now, I wanted to look at a different question. And that question is, what did this thing come from? Where did this virus come from? It showed up in people in November or December of last year, and it spread around the whole country. And it's caused, obviously, a great deal of suffering, people dying, and illness, and a shutdown of the global economy. So it's pretty important to figure out where it came from. So I'm going to just go into this link, this link to the entry in another database, in a database called GenBank, which is what NCBI nucleotide is searching, gives a lot of detail. Now, I don't think you need to understand all of this, but this is the entry this has details of those proteins that are predicted to be made by this virus, and then the virus sequence in nucleotides from the start at 1 all the way down to 28,881. So, it's almost 30,000 nucleotides long. That's pretty small compared to the 3.2 billion in the human genome. But it's got a lot of information in there, and it has these proteins which help the virus exist and make copies of its RNA. Okay, so I won't go into that much more, but what I want to do is try to figure out what this thing might have come from. And to do that, we can use a search tool called BLAST. So I just want to go back to my PowerPoint just briefly, because you may never have heard of BLAST. So BLAST is Basic Local Alignment Search Tool. It looks for regions that are similar between biological sequences, and they can be nucleotide or protein sequences. Okay? I'll go back to BLAST in a second. There are different flavors of BLAST. Because nucleotides are similar, DNA codes for RNA. And because DNA and RNA code for protein, you can do searches of DNA with DNA, or of DNA with RNA, or vice versa. And you can also look for nucleotides with a protein sequence, because if you know the amino acids, you can figure out what the possible nucleotides that code for them are. And that allows you to search in between proteins and nucleotides, okay? Now, for now, what I want to do is I want to use the nucleotide sequence of this virus to look and see what we can find, okay? So I'll go back to my hits to this Wuhan HU-1 complete genome, the first of the genomes that were ever looked at. And I'm going to run this program called BLAST, which uses the nucleotide sequence to look for other nucleotide sequences that are related to it. Okay, and the search tool gives bonuses for things that match well. Things that are identical are the best. Things that look kind of the same are a little worse, as you might expect. Okay. So I'm going to run this program called BLAST. Now it automatically loaded the number NC045555512.2. That's a database number for this sequence. It's easier than cutting and pasting all those nucleotides and putting them in. Okay. Now I want to do something kind of special here. I want to look for everything except this virus, okay? So I've done something special in this BLAST search. I chose to enter in SARS-CoV-2, okay, that's the name of this virus, and I wanted to exclude it from the search, okay? So I'm not looking for other versions of this virus right now. Instead, what I'm doing is I'm looking for viruses other than this one that are similar, okay? So when I do that, what I'm hoping for are things that are related, and hopefully that'll give us some clues as to, where the virus might have come from, okay? So off I go. Now, NCBI BLAST is pretty fast, but this tool is used by people all over the world to look for related sequences all the time. So this might take a few minutes. Let's hope it doesn't take that long. But in the meantime, we can speed things up a little bit by going to the results if we need to. But I don't need to, because there it came through. All right, so here are these results. I'm going to zoom out a little bit, because what I want to show you is this stuff down at the bottom. So at the top here is some details of what I searched with. It's this SARS-CoV-2 sequence. It's 29,903 nucleotides, apparently. That's a little longer than what I read in the GenBank entry. It says what we searched in. We searched in the nucleotide database, okay? And then at the bottom here, it gives descriptions by default, and there are different tabs, and you can look at this in different ways. So the very top of the list is a bat coronavirus, which I don't know if Dr. Seuss had anything to do with this, but it's bat coronavirus rat G13. I know I'm sort of misreading that just because it sounds like it rhymes, but it's a bat coronavirus, and it's really very similar. It's 96% identical. That means that 96% of all the nucleotides, the A's, U's, G's, and C's in these RNA viruses are exactly the same. That's really very similar. And the next most similar one is from a pangolin. You might have heard of these in the news a little bit. These pangolins are sort of like anteaters with scaly armor. And there's been some discussion about them being related to SARS-CoV-2 as well. Okay, but by far the most related one is this bat coronavirus. Okay, and if we look at how they line up, it's very clear that if you look at the nearly 30,000 nucleotides from the query, the thing we searched with, our virus of interest, the top hits are this bat coronavirus and then pangolin coronavirus, which seems to have a little spot that's different, okay? It looks like there's a little bit that's not quite the same. So these are lining up the bits that match in red, and then there's a little gap for this one, for the pangolin virus, and the bat virus is the one that is the most related. But really, quite remarkably, the bat and the pangolin virus are quite similar to this thing. Now, we can also look at this in sort of a family tree or a taxonomy, and you can see that these are all viruses that fall into a family, okay? So this is looking at what kind of viruses they are, and the bat coronavirus is really the best one. It has a super high score, okay? Which means it's really similar, followed by a few other bat SARS-like viruses, SARS being another human coronavirus that caused a different pandemic. And then there are some other related viruses in civet, which is a wild cat, and in bats again, okay? So that's very interesting. It looks like maybe it came from bat. Now, I won't go into all the details, but maybe there was a bit of mixing between bat and pangolin, and there's been some discussion of snake, but we don't have time to go into all those details. But I wanted to show you that if you do these searches, you can find lots of related things, and that can be very informative, okay? Now, this isn't the only way you can do searches, and I wanted to show you another one that's related But first, let's go back into the PowerPoint and let's talk a little bit about what that genome can say. So I'm going to go back into my presentation. Hopefully I can make it full screen there. And remember, we went and we searched and we found out a lot of things about where this thing might be from. But I wanted to go back and just say a little bit about what these genes might code for. Okay, so here's a cartoon version of what this coronavirus might look like. On the very outside is the product of the S gene. It's a spike protein. It's called a surface glycoprotein. It's on the surface of the virus. And attached to the proteins are some sugars. That's why it's called a glycoprotein. Okay. There are other structural proteins, the M protein, and then a protein called a nucleocapsid protein that wraps itself around the genome, the genomic RNA. Okay. And then there's an envelope protein, which is also part of the sort of surface of the virus. Okay. Two big regions code for proteins that are important for how the RNA gets copied. These proteins form something called a replicase, which replicates the RNA. It also helps read the RNA, translating it into the other proteins. So here's a sort of pretty famous image. I think you may have seen this on the news and many other places. This is an image of a coronavirus. It was produced by the Centers for Disease Control, and it's very, very pretty, which is why it shows up so many places. I just wanted to point out that, you know, the spikes on the outside, these are these spike proteins. It looks like a corona. So the sort of fiery thing around a solar eclipse. So that's why these viruses are called coronaviruses. And I also wanted to point out that the spike protein binds to a human receptor, and that is important for how the virus gets into human cells or other host cells. Okay. So you have the virus on the surface, you have these spike proteins. I'm not going to go into it, but the spike proteins actually do some sneaky tricks. They fold themselves up to make themselves hard to see by the immune system, apparently. At least that's one theory. And but when they're near these receptors called ACE2 receptors on the surface of human cells, they can bind, and that allows the virus to then become engulfed. And eventually the RNA is released, translated into protein, and through, you know, so you get the RNA copied and the RNA read to give these proteins that then get packaged along with RNA to form new viral particles, which are released and can go on to infect other cells. Now, I know that was a lot to put into one slide, but I just wanted to have something here that gives you an idea of what is going on when this virus is infecting a cell. Okay. Now, in fact, this is a lot of very important molecular biology, and it might, in fact, be very interesting to you, and it would take a long time to explain everything in detail. Okay, so I apologize for not having time for all of that. Okay, so back to this question of where the virus came from, and we did this search with BLAST. I I didn't show you in detail, but the search results also show, on top of it being related to this bat coronavirus and to SARS, it's also related to another human virus that was also a coronavirus, MERS. So in the past few years, we've had this current pandemic and 2 other pandemics from quite closely related coronaviruses. And it looks like these coronaviruses are kind of dangerous when we get in contact with them, and they maybe mutate to survive well in people. So here's a family tree of these viruses. So if you compare all of the sequences from a bunch of different coronaviruses, you can see that the people from Wuhan who had this mystery virus, they all had very related sequence, and it was extremely related to this bat CoV RATG13. And then it's related to MERS and SARS in people, okay, amongst other things. And, you know, my favorite, and it's just because I think they're cute, is that it's related to a virus from a hedgehog. And hedgehogs are kind of spiky, and the virus is kind of spiky. I know it's a little bit silly to think about these scary viruses that way, but I like the spiky hedgehog CoV. Okay. Now, we can dig even deeper. So I want to go to this other website that's called NCBI virus, just so you can see these tools. Okay. So NCBI virus is a database, but it's not for all nucleotides. It just focuses on viruses, and it's really well organized if you're really interested in viruses. So, you know, I think everyone in the world right now is pretty interested in viruses, and this is a place to go and look. So I'm going to head now to NCBI virus and take a look in there. Okay, so here's NCBI virus. It's a pretty big database. It has many sequences in it. Something like 12,000 reference nucleotides. So those are nucleotides that are the sort of reference for a type of virus or a variant of a virus. It's a lot, okay? But the tool is really nice. You can do searches in various ways. Okay, you can search by sequence, you can search by virus. Okay, so you can do a search just with the name of a virus, or you can do a search with a nucleotide sequence. And, you know, the way we got into this was with a search of NCBI nucleotide, and we got a result for something that came from a virus in Wuhan. And just to remind you, that was this thing, NC0455, et cetera, okay? And NCBI virus understands that code just like NCBI BLAST. So I can do a search, okay? You might also notice that there's a search for up-to-date beta coronavirus database, DB for database. That's a search that's specifically just for beta coronavirus, which I think has been added recently just because of this pandemic. Okay, so I'm going to do a search for this nucleotide. And it's searching using BLAST, okay? So it's looking for things related to that original sequence. Now, this is pretty complicated, but it's got a lot of really cool tools that you could explore on your own when you have more time. You need a browser. You need to remember it's NCBI virus. And then you can do lots of searches in here. For instance, you can click on this plus here by virus, and you can search with a keyword like SARS or MERS or COVID-2. I think you'd find things that way, okay? We'll come back to that, I think. I want to make sure that I'm only looking at sequences that are complete, so whole viruses. So I'm going to the one tab here to refine my results by clicking complete. Okay, and it says I have 445 viruses. And you can see that they're from many places, from China, from different parts of the United States, Korea, Florida, Georgia, from all over the place in the world. These are places where this virus has been found, okay? And you can also sort them by date, and you can see that scientists have been searching for these viruses systematically through the time since it was discovered, okay? Now, these are blast hits, so they're not all 100% identical, and in fact, some of the blast hits aren't very identical, right? The ones that are, and I'm just clicked on identity, the ones that are really highly identical are almost all certainly this same virus, but with mutations, okay? So turns out that viruses often mutate a lot, and that's because when they copy their genomes, their replication machinery, or the way they get replicated within the cell, is a little bit more error prone than our genome replication machinery, which is very good at replicating DNA with few errors. And that is important to these viruses. It allows them to change rapidly, and they evolve over time, okay? So anyhow, there are all these hits, many, many hits, and you can see that they're not all the same. And if I click this little button here, I can select all of the hits that came up, 445 of them. It's really kind of daunting. So if we wanted to try to go through all of that all at once, I think we'd drown. And in fact, I'm sure many of you are a little surprised at the amount of information, but science has been really doing a lot of hard work on this because it's such an important thing. It's critical that we figure out as much as we can about this virus, okay? Now, NCBI virus has some tools built right in. So you can take all of these sequences and you can line them up, okay? It uses an algorithm which looks for the similarities, and then it highlights the differences. And if you have followed any of our other presentations from the Learning Center over the last month or so, you may have seen Jason Williams or others comparing sequences in the blue line of DNA Subway, or in a tool we have called Sequence Server to compare human sequences or sequences from different organisms. Well, you can compare sequences of viruses too. So I'm going to just go click on alignment, and this will launch an alignment of all of these viruses. Now, 445 sequences is a lot, and it takes a long time for the computer to do this. Imagine all 445 sequences, all of them 30,000 long, have to be compared to each other and then lined up, highlighting the differences and the similarities. So this takes a little bit more than no time at all. So we'll let that think for a little bit, and I'll go back to my PowerPoint. And we've done a search, and I just wanted to remind you of something. If you have mutations occurring, they occur over time, and you end up over time with a little family tree based on these mutations. So over on the left, we have a red branch. Maybe there was a mutation, one single mutation, and now the descendants of the virus would be different, and you could tell that. Maybe later there's another mutation and you get a new difference that can follow a substrain. Okay. And for this virus, because it mutates pretty quickly and it's in many people and we've sequenced a lot of versions, we're now getting some tools to look at the virus, how it's changing, but also if you look at the virus from different places and you keep track of where the mutations were first found, you can use this as a tool to track where the virus went. You know, who got the virus from whom, or maybe not exactly the right people, but the substrains can tell you a little information about where the virus is moving. And remember, these sequences, they also do things. So maybe you can find out something about what is going on with the virus and how it's changing in terms of its function, right? So scientists will use mutations to track lineages, and they also look at the mutations and try to figure out if they tell you anything about function. Both of these are really important for our ability to understand how the virus moves and how to control it. And it's also really important for figuring out the biology of the virus. Maybe some of the mutations are important. Maybe some of the mutations could affect how we treat the virus, okay? Now, it's still very early on in this pandemic, so not everything is being sorted out, but we can certainly do some of this. Okay. So we'll go back into NCBI virus. And I can show you these alignments. Now, this might be very faint on your screen. I find it quite hard to see. But here's a lineup of the 28 to 29,000 nucleotides from all these different variants. Okay, many, many, many different variants. These are all of this one virus, but in different places. And there are little red ticks where things are different from each other. So these are all differences in the virus. Now, figuring out what all of these are is pretty hard. But if we zoom in a bit, you can see an interesting thing. There are parts of the virus that maybe are getting mutated more than other parts, or at least the mutations that occur are sticking around. Okay, so what we have are viruses from individuals. These are viruses that actually can survive and replicate and get, can infect other people. We know that because they're in people now. Imagine mutations that are bad for the virus. Well, maybe we don't find those. So maybe these mutations are things that don't matter, right? It could be that it's a neutral thing. You can have a mutation and you can use it to tell viruses apart, but it has no effect on function. But maybe some of these are actually functional and could have a positive effect for the virus, or change how the virus behaves in some way. Maybe change them from being a little bit more deadly to being a little less deadly, or maybe they become more effective or have less symptoms. These are all things that could affect our ability to treat the virus or find the virus. A lot of people seem to get no phenotype, no symptoms. I guess it's not a phenotype of being infected by this virus, whereas other people, of course, have very serious consequences. Okay, so that's one way to look at it. And you can see there are many, many mutations in many places in the virus. So it has changed a lot just in a few months because it replicates very quickly. It doesn't copy its DNA very well, RNA, sorry. And, it's in a lot of people. So it has a lot of opportunity for change. Okay. So I'm going back to these BLAST results, and I just wanted to build a phylogenetic tree. So instead of comparing the sequences and lining them up, you can compare them, look at the similarities and differences, and make a little tree that shows you how the different variants are related. And you can see, I hope, that the viruses are related to each other. They're all part of this tree, with some viruses that are on these little branches that are closely related to each other, and some that are much more distantly related. So the horizontal axis here is the amount of differences, essentially. And you can see there are all these sub-branches. So mutations have occurred, and then more mutations and more mutations down each of these branches. And this provides a lot of information if you also combine it with where these viruses are found in the world. Okay, I'll come back to that in a little bit. Okay, so I know that's a lot. We have this virus. We can find it in a database. Its sequence is being published. You can take that sequence, you can compare it with everything in that database. You find some hints to other viruses. You can learn a bit about what the genes do. You can learn how the virus is changing over time in various ways. But what does this mean? Okay. So if we take a look in the virus and zoom in on a region that seems to be mutating a lot, there are in fact some really important things in there. Okay. Now I'm going to switch from something that I did and showed you in NCBI virus to something that a researcher did and published, because they dug into it in a great deal of detail and then looked at what they think might be going on. Okay, so I'm just going to focus on one part of this one paper, and there's a lot of research on this, but I thought this was a good example of something that maybe people could understand. So remember, the S gene codes for the spike protein. Okay, and in the spike protein is a region which is the receptor binding domain. So domains are parts of proteins that fold up and do a function. And this particular part of this spike protein on the outside of the virus is thought to bind to this receptor. Remember, I called it ACE2 earlier. Okay, and in this region, there are mutations right in this receptor binding motif, the part that actually binds to ACE2. Okay, there are several mutations in there, and that seems pretty fishy. That seems like, this could affect how this virus binds to a human protein important for how it infects the virus. And that seemed to these people like maybe there was some evolutionary reason for that. Maybe there's some selection going on. So if you have a random mutation in this area that affects the amino acids in the protein coded for by this gene, maybe that would change how the protein works. Now, they haven't gone on to actually figure out what the effect is, or if they have, I didn't have time to figure that out properly. But they did look at, computer modeling to figure out if there was any selection. And they think that this region is under positive selection, meaning that some of these mutations actually are helping the virus. Okay. And if we look at the structure of the interaction of ACE2, which is in green up at the top here, with the SARS-CoV-2 spike protein, the region where these mutations occur is right at the junction of the receptor and the protein, okay? So I think almost anyone can understand that if you have two things that are interacting with each other and you have changes in the structure at that boundary, it could have a really important effect on how those two things match up, right? And we don't know the implications yet, but it looks like this virus is mutating, and some of the mutations are, at least according to this one group, important for the virus. They're actually helping the virus, okay? And so they're selected for, you know, that's one thing you can learn from looking at genomes and comparing them, in this case, within this one virus and its sub-lineages, okay? So I thought that was a pretty interesting story. So the other thing I wanted to talk about, and I realize that time is moving quickly, is how these mutations can be used to track viruses. So remember, you can have mutations, you have these sublineages, and once you have a mutation, the descendants will inherit those mutations. Almost always they don't mutate back, although that can happen. It's called a back mutation. But usually you can just follow the lineage. You have mutations down different lineages. And if you know where the people or the organisms or the viruses came from around when those mutations occurred, and then you find them somewhere else, chances are that mutation came from that original place, right? You know, it is possible to have the same mutation happen more than once, but by tracking multiple mutations, you can track where a virus goes. So I want to show you one tool that's been developed that takes all of this information and puts it all in one package, because I think it's really a cool example of taking a lot of data and visualizing it in a very powerful and I think actually quite beautiful way. Okay, so I'm going to take you just at the end here to something called NextStrain, which is a site that tracks different viruses, not only this virus, and shows patterns of where viral strains have gone in the world. Okay, so it's a lot of information. I don't think you have to understand everything, but maybe you'll see the extreme power of a combination of bioinformatics, comparing sequences, and then tracking these variable sequences in time and space to get a pattern of what is going on. Okay. So I'll try my best to navigate my way into NextStrain and show you that. Okay. So NextStrain, if you do a search for it, it's NextStrain with no spaces. And the site lets you look at a lot of different viruses. And you can plot a bunch of different things in here over time, okay? So it's a pretty confusing site in the sense that you have a lot of things going on. So the screen off to the left has things that you can play with. You can play with them yourself if you go there. You can change the shape of the family tree. There are lots of ways of looking at family trees. Some are more straightforward and look like a family tree, like the default, which is this rectangular version. Some are a little more compact and kind of highlight the differences and similarities, like this circular one. Some of them put time in there, so you can track the trees as they evolve over time, which is the version that I like to look at. It shows that, it kind of shows how things change, and you can watch them change as you move through days, okay? So you have the tree, you have a map of the world. At the start, this started in China, so it's just in China. And then at the bottom is a map of the genome. And at the start, there's nothing really interesting going on there. Why is that? Well, because this is all about the genome and changes, and you can have it set for amino acids or nucleotides. But remember, nucleotides code for amino acids, so they're roughly equivalent, at least at the level of detail that we're going to look at, okay? So here's the cool thing. You can do all sorts of stuff. You can change where you're looking. You can do it by country. You can do it by state or location, okay? We'll just stick to country for now, okay? You can color by which country? You can color by genotype, that's the strains, okay? And that allows you to look at different aspects of this data in the screen, okay? So I'm gonna hit play here, and maybe some of you have seen this before or seen part of this before, but I wanted to show you the tool so you can go there and you can do all of this. Fiddle with it. See what you can see. See what your brain picks up. You can pick up a lot of patterns. So here, I'm going to play it. And over time, you'll see that the virus is going to migrate out of China. And while it does this, it mutates. So first it mutates just in Wuhan, and then it mutates and moves to different parts of the world, right? So early on, it's just in China, then it migrates out into countries near China, or at least as far as we knew. And eventually it hops into Europe, in the Middle East, and North America. And then from each of these places, it starts hopping around back and forth. And why do we know this? Because you can track different mutations, okay? And over time, more and more mutations accumulate in different parts of the genome too, okay? So hopefully, as this finishes, you can see that by looking at genomes and comparing them, you can figure out where a virus might have come from, what creature it was in originally, how it changes over time, what it codes for, and where it goes and mutates as it's going, okay? Now, of course, this all starts looking like a big scribble, but it's quite remarkable how the virus has changed a lot. And there are, many mutations. There are viruses that have accumulated over 20 mutations in a substrain. Okay? So these are the number of mutations on the left in this graph. Okay? So I hope you enjoyed that. I hope that my colleague Sharon Pepinella was able to answer any of your questions. And I hope that this gives you some tools. They might be a little tricky at first, but tools you could try to search for sequences, understand a little bit about these viruses and track the virus in time and space, in part by using mutations. And with that, I guess my hour is up. I'd like to thank you for joining me, and I wish you all well. Thank you very much.